Right. Okay, so I'd like to thank you for coming to the session on antibody engineering of drugs for new treatments. We're going to start with a presentation by Professor Nissim, Ahuva Nissim. Ahuva graduated in molecular immunology in 1992 from the Weizmann Institute of Science, and then she went to do a postdoc at the MRC Center for Protein Engineering with Sir Greg Winter, who we already heard a great overview of antibody engineering and so on earlier this afternoon. And Greg, as I think many of you know, was awarded the Nobel Prize in Chemistry in October 2018. During her postdoc, Ahuva built a phage display semi-synthetic human antibody library, the so-called Nissim library, that's become widely used in many different countries and been a huge asset, actually, for the library, antibody library work that's been ongoing in many laboratories and biopharma and so on. So basically, she joined Queen Mary in 2000, and her research there has been focused on the mechanisms that lead to the formation of these disease tissue-specific neoantigens and how that might be used, how these neoantigens might be used as diagnostic and therapeutic targets. So without further ado, Ahuva.
Hi, thanks, Sally, for the introduction, and I would like to thank the organizer for letting us present our work. We will have in that session 4 speakers, if I can get 4 speakers. It will be myself first, then Professor Sally Ward, which is Director of Translational Immunology in University of Southampton. Then it will have a break with oral communication. Then we will have Professor Rene, Chief Innovation Scientific Officer in FairJourney and Professor of Biopharmaceutics at Maastricht University. And then we'll have Dr. Gary Mary from Twist Bioscience. What I would do in my talk, I will take some slot of my time to give you an overview on antibody engineering, so we are all in the same page. So the whole thing started back in 1975 in the discovery of the hybridoma technology by Kohler and Milstein. Some of the slides that I'm showing was already shown by Sir Greg Winter, but I'll go quickly so the audience that were not in Sir Greg Winter talk will be with us. So the problem here was using mouse monoclonal antibody to treat human is a problem because the human were responding against the mouse antibody because they recognize it as a pathogen. So there was a lot of effort in antibody engineering to minimize as much as possible the antibody epitope. So the first attempt was to make a human mouse chimeric antibody where the antibody variable domain was transplanted into the Fc of human antibody. They are still in the clinic, the rituximab, the anti-CD20 monoclonal antibody that are used to treat malignancy and rheumatoid arthritis, and the infliximab anti-TNF for the treatment of RA. The next approach, which was also described by Sir Winter, is to do a humanized antibody where the loops in the antibody that make the direct contact with the antigen from the mouse monoclonal antibody was transplanted into human backbone, and this is the humanized antibody. So you can see much less mouse epitope in the antibody backbone. The first antibody that went into the clinic was the anti-CD52, which is used until now for transplantation, autoimmune disease, and now most likely it is used quite a lot in multiple sclerosis. And the other example that I'm giving is the Herceptin that is used to treat breast cancer. So the display technology, as described by Sir Winter, was a big game changer. So it's actually using a genetic display package to have the antibody gene from the inside and displaying the antibody protein on the surface of the phage. So this is actually mimicking the B cells that have the gene for the antibody inside and the antibody display on the B cell membrane. So the most common technology, as mentioned by Sir Winter, is the phage display. But in parallel, there are other technologies that were developed alongside, is the yeast display, and more recently, the mammalian display that will be covered by Professor Hoet. So the type of antibody repertoire that we are using is either using a human repertoire for a gene from human donors, and what Dr. Mary will show to you actually, that we can use a phone, like a chip a little bit larger than a phone chip to make the whole antibody repertoire artificially. And this will be covered by Gary. So once you have the repertoire, you display it. Most commonly, all of us is using the phage display. You can display Fab, single chain or single domain. And then once you have performed selection, you readily can convert the antibody fragment either to intact antibody or to antibody fragment, whether B-specific component like attracting effector function together with binding as a fragment or as an intact antibody, and or linking to the antibody some treatment, what is called antibody drug conjugate, ADC, or making a chimeric T cell. So the engineering doesn't end up by engineering the variable domain. There is a lot of engineering on the Fc part of the antibody to engineer the binding to the Fc receptor, to increase ADCC and CDC, or also playing with the carbohydrate, not just with the amino acid, again, to modify the ADCC and CDC. And what Sally will cover is actually the modulation of the FcRn receptor to modulate the half-life of antibody in the circulation. So as you can see, the antibody which are in the clinic, there is now about 131 antibodies. And you can see the number of antibodies that are used in the clinic keep increasing, both for non-cancer application and cancer application. And you can see that more and more antibody format, not just a full antibody, are becoming more available for clinical application. So this is the introduction that I wanted to give to you. And now I'll go to the topic that I would cover in my talk, is actually we still have a challenge to choose the correct target to deliver drug. So, and in my talk and my work, actually, I was putting a lot of effort to identify the optimal neoepitope, which can be used to target drugs specifically to the disease tissue and minimize the side effect. So I would be giving you an example from musculoskeletal disease and type 1 diabetes. So musculoskeletal disease, we were focusing on rheumatoid arthritis, which is a disease of inflammation in the small joint, mostly synovial membrane inflammation. We did some work on spondylitis arthritis, which is axial, or psoriatic arthritis, which involve all the skin. And also we did some work with osteoarthritis. So when we look inside into the inflamed joint, apart from producing high level of pro-inflammatory cytokine and matrix metalloproteases as a result of the inflammatory process, there is a high influx of immune cells that penetrate the inflamed joint. As a bystander, they produce a lot of oxidant, starting by superoxide, that is immediately converted to peroxynitrate, H2O to OH or HOCl. So when you look at because we are dealing with a joint, what was interesting to us to look at a major component that can be attacked by this oxidant. And because collagen type 2 is the major component of the cartilage, so we use collagen type 2 as a protein to validate our hypothesis. So what we did, and this is the step that we use, we take collagen, expose it to oxidant, and then first of all, we check human sample to see if there is any reactivity against this protein. Once we have an answer, which is yes, then we develop antibody against this oxidized neuroepitope. We do some in vitro functional validation of the antibody. Then we do some imaging study, then we develop antibody-drug conjugate, and then we do another in vivo experiment to see the efficacy of this antibody-drug conjugate. So this is to check that there is reactivity in serum sample from rheumatoid arthritis patient. You can see very high level of reactive oxidant-specific antibody in blood of arthritis patient. We did see a little bit sometimes when a patient have only pain but not arthritis, but mostly negative. Some reactivity in OA, but this is healthy control. In spondylitis arthritis, we see high level of reactivity, of IgG reactivity against oxidized collagen. This is the control RA again. But we did not see IgG reactivity in psoriatic arthritis, but we did see high level of reactivity of IgA against oxidized collagen in psoriatic arthritis, axial spondylitis arthritis, but not in RA. Having this information in mind, we use the phage display technology and develop antibody which is specific to oxidized collagen, and we see here very nice staining of the damaged RA cartilage by the antibody, opposite to staining with staphonin O, we stain only intact cartilage. This antibody doesn't stain healthy cartilage, and this is a control anti-native collagen antibody that does stain the healthy cartilage. Very nice staining with staphonin O. This is in vivo imaging study, very nice localization of the antibody into mouse model of antigen-induced arthritis, where you induce the arthritis in one knee and the contralateral is healthy. This is antibody fragment, half-life of 3 hours. This is intact antibody, and you can see localization of the antibody into the inflamed knee, no localization with control antibody. Then we fused the antibody to drug. The first fusion we did antibody etanercept, and we could see again in mouse model inflammatory arthritis a significant acceleration on the disease and resolution of inflammation. And also when we fused the antibody to IL-10, significant acceleration in disease resolution. And here we actually used particles, and we loaded on the particle both anti-TNF via IL-10 and the anti-oxidized collagen antibody, and you can see really very nice adding up of effect when we have both treatment. So we did some work also in osteoarthritis, very nice staining on the damaged area of the OA. And this is the severe OA, this is mild OA. Nice staining around the chondrocyte, which is the cell that populate the cartilage. And most likely these cells are trying to resolve the damage which is here, and that's why they are active metabolically. This is imaging. That's another type of work that we did. And you can see very nice staining. Even there is no defect, we already start to see staining with the antibody. So the antibody is really very good in very early detection of OA. And the more damage, the more staining. And this is in vivo imaging, localization. This is the DMM model where you cut the ligament here, and slowly the mice develop OA. very nice localization of the antibody, no localization, which is just a sham operation. So we wanted to have an example of our concept in a completely different disease with a different issue. So we went study type 1 diabetes. This is a more recent study. This is a disease of the pancreas. Beta cell in the pancreas is slowly getting destroyed. Level of the glucose is going up in the blood. Level of insulin is going down. So in the gold standard diagnostic of type 1 diabetes is for a target autoantigen to which children develop antibody, GAD, insulin, tyrosine phosphatase, and zinc transporter protein. And the first antibody that is detected in children is the antibody to insulin, even before dysglycemia, meaning imbalance of the glucose in the blood. So what we did, similar to what I showed you with the collagen, we exposed the insulin to oxidant, and we saw a very high reactivity of serum sample from children with type 1 diabetes against the oxidized insulin, not so much against the native insulin. Type 2 diabetes, we saw a little bit, but definitely not as much as type 1 diabetes. And we took two healthy control, school children for that group, and adult for the type 2 diabetes group. At that time, we had a lot of argument with the reviewer because the concept of being able to do ELISA with insulin was not there. So we needed to do the same sample that were tested here by our ELISA. We needed to do also radioimmunoassay to confirm the result. And we have 50% correlation between the two assays. We were detecting another 34%, and the radioimmunoassay were detecting another 11%. So altogether, we could detect 95% of the children. So in the next study, we wanted to see whether the reactivity against the oxidized insulin is ongoing also before the onset of the disease. And yes, we took children between 11 and so years before the onset of type 1 diabetes. And only the children that develop type 1 diabetes respond to the oxidized insulin, but the children that did not develop type 1 diabetes were negative to oxidized insulin. More recently, we wanted to identify the exact epitope in the insulin to which the serum sample are responding. So what we did, we exposed the insulin to the oxidant, and we sent the oxidized insulin to mass spec analysis. And what we saw is we identified the exact amino acid to which oxidation is happening. The next step, we expose insulin to oxidant, and then we subjected to size exclusion chromatography, and we took each separate peak and tried to see which are the peak to which serum sample from type 1 diabetes children are responding. And the one, so we took all these peaks, we did ELISA, and the peak that shown to be reactive was taken again to mass spectrometry to identify the amino acid sequence in the peptide that are composing these active peaks. And we have identified 6 different peptides from the insulin sequence. There is another version of peptide 6, which is with and without the R, and the R is actually the amino acid between the insulin and proinsulin. So, so we synthesized all these 6 peptides. So peptide 3 was from the alpha chain, peptide 4, the longest peptide from the beta chain, peptide 6 from the beta chain, and so we had another 3 peptides that were not active, so I'm not going to talk about them. So the first thing is to synthesize this peptide expose it to oxidant, send it again to mass spectroanalysis, and validate that indeed we oxidize the peptide by monitoring the mass of the peptide. You can see the change in mass here between 701 to 457, and the same for peptide 3, one peak here, and you can see several peaks after modification. So once we confirmed by mass spectrometry that indeed the peptide is oxidized, we went back to ELISA, and actually we could see very nice reactivity of type 1 diabetes patient to the oxidized peptide, whether it's peptide 3. With peptide 4, we didn't see so much difference between native and oxidized, and we think that this is happening because peptide 4 is very long, has cysteine, probably it's auto-oxidized by the time we are measuring the reactivity. Peptide 6 is very nice again, very high reactivity in type 1 diabetes children against the peptide, not so much in the healthy control. But we wanted to be more convinced, to be a really more striking result. So what we did is actually, we took the serum sample from patient and incubated that with the peptide, whether the peptide was oxidized or native. And then we put it on a ELISA plate that had either native peptide, native insulin, and oxidized insulin. And what we saw is actually when we incubate the serum sample with the oxidized peptide, we inhibit the binding to the oxidized insulin, not to the native insulin. This is for peptide 3, and quite striking for peptide 6. You can see inhibition here. But for peptide 4, the inhibition was done by both native and oxidized, and the effect was not as striking compared to peptide 3 and peptide 6. So when you take a peptide and oxidize in the test tube, you can get variation. If you do it today, or if you do it in a week time or 2 weeks time, you will not get the same batch. So we decided to oxidize the peptide by designing in silico oxidation. So looking at the peptide sequence, for example, phenylalanine, which is in that sequence, can be converted to tyrosine by adding the OH here. And the tyrosine, which is here, can be converted to DOPA by adding another OH. So, for example, in this peptide, we had few diversion of the oxidized peptide when we have the YY, while we are converting only the phenylalanine, but we have another version that is converting the DOPA, the tyrosine to DOPA. So that is the 4 different peptides, naive and modified. And for peptide 3, on top of the tyrosine that were converted to DOPA, we also have the cysteine here that were converted to cysteine by oxidizing the tyl group here with 2 oxygen and OH. So once we have this, we went back to the mass spec, and you can see changes in the mass when it is native compared to the oxidized. This is similar for the other peptide, changes in the mass. So once we confirm with the mass spectrometry that indeed the in silico oxidized peptide is showing that they are having different mass, meaning oxidized, we went back to the ELISA and again saw a much higher reactivity to the oxidized peptide in the type 1 diabetes sample, not so much in peptide 3. We did see also a reactivity to the native, but with peptide 6, it was quite clear that the reactivity against the oxidized peptide is stronger than to native peptide in type 1 diabetes. But in any case, always the reactivity against the peptide was higher in type 1 diabetes compared to healthy control. We wanted to see whether this phenomena is specific to the humoral response and whether T-cell response, there is a cellular T-cell response against this peptide. So we exposed a peripheral blood lymphocyte to the different peptide, and surprisingly, we saw that the dominant epitope for T-cell stimulation is in fact peptide 4, both for CD4 and CD8. Then we say, okay, this is a different cohort. Maybe there is a variation between one cohort to the other. So we check this cohort again for the IgG response, and in fact, peptide 6 again was the stronger peptide in terms of reactivity of serum sample from type 1 diabetes. So we sent the peptide to our colleague in Seattle, and we wanted to see whether the T cell response is naive cells or memory cells. So if you compare it to control influenza stimulation, most of the response is memory cells, very small portion is naive. The naive portion is much higher for the insulin, but you still have a memory T cell response. This is a very preliminary experiment that we need to validate further. We did some Venn diagram, and we could see that we have about 40 to 80% concordance between T cell response and antibody response. And when we did a PCA analysis, we could see that most of the sample that we are testing for the T cell reactivity are grouping out, and the antibody reactivity are grouping out from the healthy control individual. So if we go back to what we were doing is, so we took the insulin, exposed the insulin to an oxidant, identified the peptide that were resulting after oxidation, did in silico modification of the insulin peptide, and what we saw that peptide 4 is the one that mostly stimulating T cell, while peptide 6 is mostly stimulating antibody response. So I think that this is going to be quite interesting. First of all, one arm of that discovery is actually to identify a neoepitope that, as I showed you with the insulin and collagen, and use this one to diagnose reactivity in serum sample. And then once you have the antibody, the recombinant antibody by phage display or yeast display or mammalian display, you can use this antibody to detect early, a very early circulation of this neoepitope. And as I show you in the OA study, we can use the antibody for early detection of OA, which is now is really a big problem because the OA is detected by X-ray, and you can detect only OA only when there is really very advanced OA and you can see evident cartilage damage. You can again, you can use antibody that is specific against this neoepitope, fuse it to treatment, and hopefully more and more study will come up, and we will show that it will go directly to the disease tissue and will minimize the unwanted side effect. And what I was trying to show you, I made it very short, is with the particle, is we could load the antibody on the particle, and we could, on top of the antibody that is functioning for targeting, we could load multiple drugs inside the particle or in the particle envelope, and the drug was very effective, much more effective than single treatment. So by that, I want to thank the founder, the JDRF that found all my study on the insulin, Bar Charity that fund most of my antibody work, Queen Mary Innovation, MRC, and Arthritis Research UK. I want to thank Chiara Vinci and Rocky Stolo that did most of the work on the type 1 diabetes, Chris, Hugh, Birne, and Han that did a lot of the work on the arthritis project. And I want to thank all my collaborators across the globe, to be honest, that helped me with this project. And I thank you for listening. That's it.
Thank you very much, Ahuva. So are there any questions? I have a question actually in persistent inflammation, say in tumors and things like that. Sorry, can you hear me? Yes. Yeah, okay. So in cases where you have persistent inflammation in oncology and things like that, so do you get this, are these neoepitopes relevant there, Ahuva?
I think yes, actually. I think this whole study is an example that can easily be implemented to the oncology because there is inflammation. I'm sure that there is post-translational modification out there, so I think it would be very, especially in oncology, a lot of the markers are self-proteins, and it would be nice to see that this self-protein is modified somehow, which is not present in healthy tissue and will be present only in the diseased tissue.
Okay, thank you.
Okay.
So Greg.
Presumably this, the oxidation is occurring all the time to very low levels. Do you think that's likely or not?
That's what? Sorry?
Do you think it's, I'm trying to work out a model by which the disease progresses. So for example, if we think about, are you imagining that what happens is when you generate these autoantibodies, that they then come along and you get more, because they bind to that bit of cartilage, you get more reactive oxygen species and the thing becomes a general generative process. Is that what you think is happening?
Yeah, I think it's a vicious cycle, yes, once it's done, yeah.
So if it does that, then presumably if you were to inject a mouse with antiserum, which was against those peptides, the chemically modified peptides, then you might get a disease accelerating due to that particular kind of thing, compared with using just straight peptides.
How about induction of tolerance? Isn't that people that are actually injecting the pathogen to induce tolerance? So you can, it depends on the dose, so maybe you can induce tolerance by injecting that peptide?
Well, that's also another possibility, but it sounds to me like it would be worth playing with your, with your peptide as a vaccine to see what actually happens, because it could be a very useful animal model. Or it might tolerize, which itself would be, so it seems to me you win by the way, you prove what you've been thinking about, your vicious cycle, and maybe you can find a way of dealing with it.
Yeah, correct, thanks.
Okay, well, Rene, you have a question.
Very nice talk, Ahuva.
Thanks.
A question a bit in direction with Greg was already touching on. Did you look at the, let's say you saw a very nice response against this oxidized peptide? Sorry?
Yeah, that's better. Yeah.
Is it better now? And so my question is, did you look at the total immune response against insulin, basically, and oxidized insulin from these patients, basically? So what is the, what's the, just to understand, what is the epitopes recognized on the insulin? Because you focus now on this one peptide, but it would be nice to understand what is the contribution of this one, let's say, a few oxidized peptides versus the immune response against insulin, oxidized insulin. And a question related to that a bit, did you look at if there is any relation with HLA matching? Maybe use the microphone.
Yes, in our first study, we did some HLA correlation, but I think we need to do it properly, but there are definitely correlation with HLA, yes. I don't want to say something that I don't remember accurately, but we did have some HLA association, yeah.
And about the epitopes recognized by these antibodies from these patients, you could perhaps look at what are the antibodies recognizing the specific peptides and what percentage of antibodies would recognizing a different piece, maybe of the
in a different what?
different piece of the oxidized insulin?
So, I mean, I'm not sure I understood your question, but if you're asking me about what are the patient recognizing, I think I showed that the patient is recognizing the peptide sequence,
Yes.
not just the whole insulin, oxidized insulin. So, I mean, one of the things that we haven't done, for example, is if we put all the oxidized peptide in one go, what will happen? I don't know yet.
Yes.
I haven't done it yet.
That was more or less my question. So
Yeah, I haven't done it yet. Like meso, like no, we haven't done it yet. Yeah.
Okay, thanks.
Okay, thank you.
Okay, well, thank you very much, Ahuva. So, move on to the next.
Okay, so now I'll take the chair head again. I want to introduce Professor Sally Ward. She will present in her talk. First of all, I want to introduce her bio. She was graduated in Cambridge, then she joined Sir Greg Winter lab until 1920, where she moved to Texas until 2018. And then she was appointed in Southampton. The big discovery of Sally is to identify that FcRn receptor regulate the level of IgG in the body, and that this can be engineered to modulate the half-life on antibody in the circulation. And more than that is actually this engineering can be engineered in such a way that it can be used to deplete pathogenic antibody from the circulation. She has developed the antibody that is now approved by FDA and now is developed by Arganex, a VYVGART antibody that is used for the treatment of multiple sclerosis. Sally, it's all yours.
So thank you very much, first of all, Ahuva, for the kind introduction and also for inviting me to speak in this session. So what I'm going to do is actually give a sort of historical sort of overview of our work related to FcRn and how that's led to the development of therapeutics in various areas. Mainly, I'm going to talk about treating autoimmune disease, in fact. So, as Ahuva mentioned, I moved back to the University of Southampton fairly recently, but prior to that, I was in Texas for many years, in fact, and basically nearly all the work, or essentially all of the work that I'm going to present today was done in our Texas lab. So when I first moved to Texas after doing a postdoc with Sir Greg Winter in Cambridge, where we were working on antibody repertoires and making V regions specific for antigens, I decided to try and work on the other end of the antibody molecule, if you will, and try and identify what regulates the dynamic behavior, in other words, the persistence and distribution of antibodies in the body. And this is not only important for normal humoral immunity to combat infections, but also the effective delivery of antibody-based therapeutics. We know that if we make an antibody-based drug, if it doesn't deliver with the right PK properties or to the right site or whatever, it's never going to be useful as a drug, even if it has really great behavior in vitro. And also we thought if we understood these processes in the body, we might be able to modulate the levels of pathogenic antibodies to treat disease such as autoimmunity. So what we did in the sort of, and I'll just get the pointer basically to try and explain this, what we did was identified FcRn as a global regulator of antibody levels and transport in the body, and antibodies of the IgG class, which are the major class of therapeutics, in fact, of the IgG class, as we've heard earlier from Greg Winter's talk. But what we did was we identified the site of FcRn binding on an IgG molecule and identified the residues, in other words, with which FcRn interacts on the Fc region of the IgG. And it was known at that time, actually, that FcRn, N stands for neonatal, it was known in the early 90s to be the regulator of maternal transport of IgG from suckling rodents or to suckling rodents from their mother's milk across the intestinal gut of the neonatal mice, or rats in fact. So its sole function was believed to be in that process of delivering IgGs from mother to young, and that was why it was called FcRn. But in fact, we found it was expressed throughout adult life, and it played a role in regulating antibody levels through its transport function, as you'll see in this schematic. Through its transport function, it can regulate antibody levels in the body and also regulate antibody transport across cellular layers. So as I say, we mapped the site for FcRn binding on an IgG-Fc. We noted it was expressed in many different cell types, and subsequent to that early work, other people have identified it expressed in hematopoietic cells, for example, as well as endothelial, epithelial cells, monocytes, macrophages, DCs, and B cells. And pretty much every cell type you look at, actually FcRn is present. It's not just in the neonatal gut of suckling rodents. But it also actually regulates albumin levels, and albumin is another ligand for FcRn that binds to a different site to IgG. But basically, so FcRn can transport both albumin and IgG within cells, and the function in albumin homeostasis was identified after we identified it as the global regulator of IgG in something like 2003 or so by Clark Anderson and Derry Rupenian. So what does FcRn do then? Basically what happens is, because the binding of FcRn and IgG, at least with natural IgGs, is highly pH dependent, with reasonable affinity at acidic endosomal type pH, but very low affinity at near neutral pH, what FcRn can do is IgGs go into cells because they can't bind, in most cell types at least that are bathed in near neutral pH, IgGs can't bind to FcRn at the cell surface. So they go into cells by fluid phase pinocytic processes. They intersect in endosomes with FcRn, whereupon they can bind. And if they bind, they get recycled back out of the cell, or indeed even transcytosed or transported across the cell if it's an epithelial cell barrier, for example. And so essentially what happens then is FcRn acts as a homeostat, because as soon as it becomes saturated, the IgG can no longer bind, and when it goes into cells, it ends up in lysosomes, essentially by default, because it doesn't get recycled back out in these vesicular recycling compartments that bud off from the sorting endosome and basically deliver the FcRn and IgG to the extracellular space. So this mechanism has been seen in real terms. We've spent a lot of time in the lab understanding the cell biology of FcRn, and what we did in an early experiment in around the early 2000s was to take FcRn, GFP, transfected into endothelial cells, and this is an endothelial cell transfected with FcRn GFP. We then see in these cells circular structures that are in fact these sorting endosomes, and essentially what we see is that if we add a wild type fluorescently labeled IgG to these cells, it basically binds to the limiting membrane of these sorting endosomes. As you can see here, these are individual frames from a movie that I'll play in a minute. And here's another endosome with IgG and FcRn localized on the endosomal structures. But by contrast, if we make a mutant called H435A that doesn't bind to FcRn, what we see is the mutant in fluorescently labeled form sits in the endosomal vacuole, if you will, doesn't get sorted out by FcRn. FcRn leaves, and ultimately what this is a late endosomal structure. We know this from our cell biological experiments, where the FcRn has already all left and the IgG is destined for lysosomal degradation. So basically this shows in a at the cellular level at least, how FcRn sorts IgGs if they bind to it away from lysosomal degradation versus ones that don't bind to FcRn and end up in the lysosomes in cells. And what actually happens then is, if I play this movie in a minute, what you'll see is in real time, this is IgG wild type coming off a sorting endosome in a tubular vesicular structure with FcRn, and this transfers or translates to a long in vivo half-life of the wild type IgG and good transport across cellular layers. But by contrast, a mutant that doesn't bind to FcRn sits, as I said before, in the vacuole of the sorting endosome. FcRn leaves without it, and basically the IgG goes to the lysosomes and has a short in vivo half-life and poor transport. So you can see at the endosomal level, therefore, the sorting processes that lead to long half-life of an IgG versus the ones that lead to short half-life of, say, a mutated IgG, or even an IgG that can't bind to FcRn because FcRn is saturated. So if we play this movie, what you see is, as I say, the tubular vesicular structure leaving the sorting endosome in that case, but in this case we see FcRn leaving without any IgG associated with it. So as I say, dynamic images of how the sorting processes go on within cells to modulate IgG levels. So we spent a lot of time understanding or defining the cell biological behavior, and by we I mean some bioengineers in the lab headed up by my husband, Raymond Ober. We've run a joint lab for many years. But basically I'm not going to talk much more about the cell biological behavior of FcRn because I'd like to now focus on modulating the dynamics of the IgG and the engineering aspects of FcRn IgG interactions that we've been involved with over the years. So what we wondered was, sort of in the early 2000s, or actually late 90s even, was if we basically engineer an antibody to bind with higher affinity to FcRn in the endosomes, but it still gets released, can we make antibodies longer lived in the bodies, in other words, now known as half-life extension? And also, can we modulate FcRn IgG interactions so we can generate blockers of FcRn function to modulate the dynamics of other endogenous wild-type IgGs in the body. And I'll show you two application areas of this, one of which has led to an approved drug, the treatment of antibody-mediated disease, and the other in the whole body imaging, which actually I'll talk about in a slightly different area where I talk about selective degradation of IgGs. So one of the things we did then in the late 90s was to take an IgG Fc region, I'm basically a protein engineer by training, take an Fc region, mutate it randomly near the FcRn interaction site, and select, and we've heard about phage display in several talks this afternoon, but basically select higher affinity variants that bound to FcRn with increased affinity at acidic endosomal pH, but still dissociate at a near neutral pH when the IgG and FcRn come back to the cell surface. And using this approach, we showed it worked in mice, and then we collaborated with MedImmune to develop a mutant called YTE, and actually we developed in our own lab a mutant called HN, or Enhance, which we licensed to argenx subsequently. What these mutation sets, YTE and HN individually, confer on the antibody is a much longer in vivo persistence. And in fact, in monkeys, for example, and even in humans, the half-life goes up 3 to 5-fold. So what this amounts to is basically a normal IgG has a half-life of about 20 days in humans. The half-life goes up to 60 to 100 days, which obviously confers very prolonged persistence of the antibody. And this has applications in multiple areas because we can take this Fc and put it on different Fab arms that bind to different targets, so that there are a wide number of different application areas one could apply half-life extension technology in. But to date, it's actually approved for several drugs used in two different disease spaces, if you will. First, in autoimmunity, Ultomiris is now approved to target complement C5, to remove it essentially, and the half-life extending mutations are used in that, developed by Xencor, in fact, called the LS mutations. And then in infectious disease, MedImmune developed nirsevimab, which is an RSV, respiratory syncytial virus-specific antibody, used in premature and susceptible infants, actually to give them very long-lived protection against what's a potentially lethal infection for these very young babies. And also, actually, in Evusheld and Sotruvimab, the SARS-CoV-2 specific antibodies, Evusheld has the YTE. It's a cocktail, as I think probably some of you know. It's a cocktail. It has the YT half-life extending mutations, and Sotruvimab has the Xencor half-life extending mutations. So after many years, in fact, somewhat gratifyingly, half-life extended antibodies are now coming through as clinically approved products. But what I'd like to spend a bit of time now talking about is a different aspect, namely, does high affinity at pH 6 and 7.4 of an IgG for FcRn generate an inhibitor of FcRn? Because what we thought was, or what we reasoned, was that if we engineer an antibody that binds more tightly to FcRn at the extracellular pH, it would allow it to be taken up by receptor-mediated processes. If we engineered it so that it also had better affinity at acidic endosomal pH, such an antibody would compete effectively for endogenous IgGs. So both at the uptake level and the endosomal sorting level, such antibodies would have huge advantages over endogenous IgGs for salvage, or salvage preference, if you will, by FcRn, essentially. So you'd drive the endogenous IgGs into lysosomes and they'd be degraded. We called antibodies of this class ABDEGs for antibodies that enhance IgG degradation. And what we did was we actually combined the YTE and the HN mutations into one Fc fragment to generate an engineered Fc, because the effects of the mutations on binding to FcRn are additive, to generate an Fc that has greatly improved affinity for binding to FcRn at pH 6 and 7.4, but nevertheless this molecule still has some intrinsic pH dependence of the interaction for FcRn with higher affinity at pH 6 than 7.4. And what we did, one of the earliest experiments we did, was we took this ABDEG for antibody that enhances IgG degradation. We took radiolabeled human IgG1 as a tracer of endogenous IgGs in mice. We injected this radiolabeled IgG into the mice, tracked it for about 3 days, and then injected the wild-type human IgG as a control, or the ABDEG containing these what we call MST-HN mutations, which is essentially YTE and HN. The nomenclature is a bit confusing, but the two sets of mutations combined. And what we saw was a dose-dependent decrease in the half-life of the wild-type IgG when we injected ABDEG at 200 or 500 micrograms per mouse, whereas with wild-type IgG at this dose, we don't see effective competition for FcRn binding, and essentially the wild-type IgG, the labeled IgG, persists with its normal half-life. So this led us to believe that ABDEGs might be quite good at clearing out antibodies in the body. And so what we next asked, our lab has had a long-standing interest in treating or understanding antibody-mediated autoimmunity. We next asked whether we could basically use ABDEGs to treat antibody-mediated autoimmunity in 2 disease models. The first one was the arthritis model developed by Christophe Benoit and Diane Mattis, the KBN model, which is a transgenic mouse. They have arthritogenic antibodies in their serum. If you take this serum and park it in normal mice, you get a very rapid joint swelling, so it's a nice model of antibody-mediated disease, or antibody-mediated arthritis. And if we did this experiment where we injected the serum into the mice, they get joint swelling measured by ankle diameter, and on day 3, when there's overt joint swelling, we inject the ABDEG, again, at about 1 mg of ABDEG per mouse. Compared to control groups, we see disease amelioration. The disease itself is self-limiting because it's a transfer model. In fact, we're transferring pathogenic antibodies that themselves clear over time. But in the control groups, which are vehicle PBS and wild-type IgG, the joints never really fully recover. You can look at the inflammatory infiltrates in the joints as well at the peak of disease in these mice, and there's a big difference in ABDEG treated versus the control groups. We also took a mouse model of antibody-mediated demyelinating disease that I'll describe also a bit later in the talk, where we essentially inject a peptide or immunize with a peptide that induces mild EAE up until about day 15 with a clinical score of about 2. This is a paralysis score, basically, in these mice. And then on day 15, we inject demyelinating antibodies, myelinated oligodendrocyte glycoprotein-specific antibodies. And again, we see if we inject the ABDEG shortly thereafter, we can ameliorate disease back to the background levels of essentially what's the T-cell response, T-cell-mediated disease in these mice. And in control groups, we don't see significant disease amelioration. So with that in hand, we felt we'd shown ABDEGs could be used to treat mouse models or treat disease in mouse models of autoimmunity. And we were sort of stuck because we were an academic lab and we didn't have the means, both expertise-wise or financially-wise, to transfer this ABDEG technology to the clinic. And fortuitously, I bumped into Hans de Haard in 2010, in the antibody engineering meeting in San Diego, and he told me about this company that they'd set up that actually at that time I had never heard of, called argenx. They had about 10 people in Belgium, and he said, we're looking for half-life extending mutations, and do you have any that you might license to us? And out of that, we licensed the enhanced HN mutant that they now have actually in phase 1 trials. But on the other hand, in the same filing or the same IP, there was the ABDEG technology. And fortuitously for us, argenx took on the ABDEG technology and, as you'll see, have developed it into a clinical product over the last 10 years or so. So what argenx did was they basically took myasthenia gravis, which is a well-known antibody-mediated autoimmune disease. It's induced or caused by antibodies that are specific, primarily actually in most patients, for acetylcholine receptor at the neuromuscular junction. MuSK and LRP4 are also autoantigens in this disease. But basically what it causes is severe neuromuscular dysfunction, essentially, and muscle weakness. Drooping eyelids are one of the first manifestations, but then leads to obviously more worsening effects over time. And it's called snowflake disease because it's very variable from one patient to another. It waxes and wanes. And basically, argenx asks the question, can we use the ABDEG, which they called ARG-113, or efgartigimod, can it be used excuse me, to treat myasthenia gravis? And what they did was essentially they took the FC region. Oh, thanks. They took the FC region of the antibody that we designed as the ABDEG, essentially, because that contains the mutations that enhance binding for FcRn, and essentially named it as efgartigimod, and then carried out clinical trials with it. And this was one of the first data sets, I think, that they produced that really indicated to us and them that, in fact, efgartigimod might lead to a clinical product, because what they did was they injected healthy volunteers and subsequently in a phase 2 study, myasthenia gravis patients, and essentially, in both cases, they saw a dramatic reduction in total IgG levels. You can actually look at the autoantigen-specific IgG levels in the patients, and they also drop. You can also look at the isotypes of the IgGs, and they also drop similarly. And essentially, this was giving 4 weekly doses of 10 mg/kg of efgartigimod. And what they noted was in healthy volunteers and in myasthenia gravis patients, MG patients, the PD effect of efgartigimod was very similar. But actually also, importantly, IgM and IgA levels, as you might expect, because they don't bind to FcRn, aren't affected. But also, actually, very importantly, the other ligand for FcRn, which is albumin, is not reduced by efgartigimod treatment, because, in fact, albumin reductions can lead to other effects like high lipid levels and so on. So this slide really summarizes a whole lot of clinical trials that argenx have successfully carried out over the recent years. The one furthest ahead, as I mentioned before, is myasthenia gravis in a phase 2 trial. They reported those results in May 2020 and showed that the efgartigimod was effective and gave patients significant benefit. And that actually led to the FDA approval of efgartigimod, which they renamed as Vyvgart, which is a more catchy name for a clinical product at least. And basically it was FDA approved, as I say, in 2020, late 2021, I'm sorry, to treat MG. Subsequently, shortly thereafter, approved in Japan in Jan 2022, and then approved in Europe. They also have multiple other indications lined up behind myasthenia gravis, for example, primary ITP and the skin blistering diseases, pemphigus vulgaris and foliaceus, and also CIDP is in late-stage clinical trials. And there are many other autoimmune diseases where autoantibodies are involved that they have in ongoing trials. So with that, it's been a wonderful collaboration for our lab, actually, with argenx over the years to see what amounted to some basic studies in mice and cell biological studies and so on, move through to a clinically approved product such as Vyvgart. But I'd like to just spend the last few minutes talking about another approach that really we see as a follow-on technology from FcRn, which I think you've all noted reduces IgG levels independent of their specificity. We began to think about ways of selectively degrading antigen-specific antibodies. And what we reasoned is if we could put an antigen on an Fc region of an antibody, then the Fc region is engineered so it binds with increased affinity to receptors on the cell surface, such as FcRn, that internalize very rapidly. We knew that from our cell biological studies. We might be able to pull the autoantibodies by virtue of their binding to antigen into cells. And we knew, again, from our cell biological studies that over time, what we call the abdeg, which is now FcRn or Vyvgart, basically goes into lysosomes over time because it basically has increased affinity at the extracellular pH. So it basically goes round and round, if you will, and doesn't get released as efficiently as a wild-type IgG would be at the cell surface. So these abdeg-type mutations, as we call them, basically end up dragging, as I say, the autoantibodies that are specific for antigen into the lysosomes in cells. So that's the first point about this molecule. The second point I'd like to make is we've ablated binding to Fc gamma receptors on these so-called celdegs for selective degradation of antigen-specific antibodies. And we've also designed the molecules that they have monomeric display of antigen with a technology called knobs into holes that was actually first described by Paul Carter and his colleagues at Genentech that makes these heterodimer structures form or at least enhances their formation. So just to briefly comment on this, because in the interest of time I don't want to overrun, but basically what we saw was that the MOG celldeg, which is myelin oligodendrocyte glycoprotein, that's the antigen component of this particular molecule that we first made, pulls autoantibodies, labeled autoantibodies into cells. We can see that in a flow cytometry experiment, and as I say, I won't spend long talking about this data. It essentially also just shows in the microscopy images on the right-hand side that again, the MOG celldeg selectively pulls antigen-specific antibodies into cells and into lysosomes. We've labeled the lysosomes in these cells with dextran, which is a commonly used label for lysosomes within cells. And in the quadrants, we've expanded regions of interest, namely individual lysosomes in the cells. Whereas the controls, which is MOG wild type, which doesn't bind with enhanced affinity to FcRn, doesn't pull detectable levels of the autoantibodies into lysosomes, as is the case for HER2 celldeg, which has a different antigen on it, which targets HER2-specific antibodies. So what we next did was we took the MOG celldeg, we basically took MOG-specific antibodies, injected them into mice, they're radiolabeled, and then thereafter injected MOG celldeg, MOG wild type, which again doesn't have the FcRn enhancing mutations, or vehicle. And what we see is actually in this experiment at the blood level, a very rapid decrease of MOG-specific antibody levels compared to the control groups. So it seems that the celldeg can clear out antibodies from the body. We also, if we look at total IgG levels in the mice, basically we don't see any significant changes with the different treatments that we've used. The celdegs, for example, at different doses, compared with vehicle, which is PBS, we don't see any change in total IgG levels. So the MOG-specific celdegs, or any celdegs, in fact, can be used pretty much at lower doses than the abdegs because we don't want to saturate FcRn in these experiments. We just want to use it as a hook to drag antigen-specific antibodies into cells, so we think we can use them at relatively low doses. And we very rapidly, as I've said, clear the autoantibodies from the circulation, and we don't seem to change the effects of non-targeted endogenous antibodies, or the levels of non-targeted antibody levels. Just to briefly comment on how celdegs can be used in PET during diagnostic imaging, and this really just shows how celdegs clear out autoantibodies in the body over time, or antibodies actually that are HER2 specific in this case. What we've done in this experiment is we've essentially injected or implanted HER2 positive tumors in mice, so this is a diagnostic imaging application of celdegs in fact. We then inject radiolabeled pertuzumab, which is a well-known HER2 specific antibody. And at 4 hours, you see distribution in all groups of the mice. You see distribution of the antibody throughout the body. But then if you inject either the HER2 celldeg, which is the cognate celldeg, if you will, it's the one that binds to pertuzumab, versus Mogg-Seldig, which is the control or vehicle, what you see over time is in the HER2 Seldig injected mice, the antibody clears out, you see it localized to the tumor. There's a lot of distribution in the liver, which I'll talk about in a minute in more detail. And basically over time you see a massive reduction in background throughout the body in the HER2 Seldig treated mice compared with the two control groups. This is the tumor on the right flank of the mice as we're looking at them. So this really is just to show you that Seldigs really do clear out antibodies at the whole body level and actually tend to take them into the liver. So the last question we asked, again stemming back to our interest in autoimmunity, is can we use Seldigs to treat antibody-mediated autoimmunity? And what we did for this was we took a model similar to the one I just described earlier in the talk when we used Abdegs, but in this case we used Seldigs. We induced a mild disease, and then on day 15, actually instead of injecting a monoclonal MOG-specific antibody, we injected antibodies from patients that had MOG-specific antibodies circulating in their blood, essentially. So these are antibodies, as I say, from what were patients with demyelinating disease, which were actually classified as MS, but probably had MOG antibody-associated disease. When we do this, we see a massive flare in disease activity in the control groups, but if we treat the mice with MOG-Seldig, following transfer of the autoantibodies, we reduce the flare, again to the level that you see without any pathogenic antibody transfer at all. So this indicates that, in fact, MOG-Seldigs can be used to treat disease in this demyelinating disease model. But also we wondered, because we're injecting essentially self-antigen in the form of MOG on an FC into the mice, which has a highly encephalotogenic epitope, we were very pleased to see that there's no enhancement of disease. In fact, there's amelioration of disease. So we therefore wondered, where do the autoantibodies go when they're dragged out of the system, if you will, by the Seldig treatment? And what we did was we injected antibody, MOG-specific antibody into mice, then injected MOG-Seldig or MOG wild-type or PBS as controls, and then by biodistribution studies, which I'm not showing here, in fact, saw that a lot of the antibody that was cleared out in the body, the MOG-specific antibody, if you will, went to the liver. And when we took out the livers and stained them, what we saw was that a lot of the autoantibodies were localizing in the MOG-Seldig treatment groups, at least, to liver sinusoidal endothelial cells demarcated by CD31 and F480 Kupffer cells, which are the sort of macrophage-type cells in the liver. But by contrast, in MOG wild-type or PBS-treated mice, we could barely detect any antibodies, MOG-specific antibodies, in the liver during these experiments. So we think that the MOG-Seldig, or any Seldig in principle, is going to take autoantibodies to the liver, which is likely to lead to tolerogenic type signals that are the liver sinusoidal, or LSECs as they're called, and Kupffer cells are known to be very good at inducing. So with that, I'd like to finish. I'd like to first thank the patients who were brave enough to participate in the phase 2 MG trials. These trials took place in many different centers. As I mentioned before, argenx ran the trials. I have no clinical qualifications. The centers were all over the world, in fact. And I'd like to just point out, actually, Chip Howard was really leading the clinical trials for the MG studies and still continues to be very actively engaged with argenx. But as you see, it's a rare disease, so that it engaged many different centers to complete the phase 2 trials, in fact. And then I'd like to just acknowledge the people who are involved in the work. Raymond Ober's my husband. We run a joint lab and have done for many years. And then these are really the key people. I've covered a lot of work, but actually these are really the key people below Raymond's name who are involved in various aspects of the microscopy and the Abdeg and the Seldig story over time. And then as argenx, we've had a wonderful collaboration for over 10 years now, with particularly Hans and Tim have really led the CSO, who actually Hans just retired, and Tim, the CEO, basically have been great collaborators with various members of argenx over the years. These are just some of the key people again, but there are many others that we're engaged with. We did the PET study that I mentioned with Zhang Kai-sun and Guiyang Hao at UT Southwestern. Ben Greenberg in UT Southwestern gave us the MS patient samples, and actually most of the mouse studies I've described with the Seldigs, or all of them at least, were carried out with human FcRn, human Fc gamma, transgenic mice provided by Jeff Ravech and Patrick Smith. We were kept financially solvent by the various funding sources listed below, and in particular, I'd like to point out the NIH and MS Society, and argenx funded essentially all of the work that I've described today. And that I'd like to just mention that we've recently moved back, as I said before, to Southampton, and we're actively recruiting, funded by the various agencies listed at the bottom of the slide. And with that, I'd like to thank you for your attention, and I'd be very happy to take questions.
Thanks, Sally, for fantastic talk. We have time for some question. Please, number 4.
Mike Parnham from Epiendo Pharmaceuticals. This was a really exciting presentation. I think probably for those who know it, it was not quite so exciting, but certainly I found it really exciting to see what possibilities are opening out. Particularly since you're taking IgG generally out of the circulation. This is of particular interest in autoimmune diseases where there are a wide variety of different autoantigens and autoantibodies. And the one that immediately comes to mind is SLE. Is anybody looking at this approach to therapy of SLE?
Yeah, so argenx have some ongoing early trials, I think it's in lupus nephritis, but actually Janssen have a competitor molecule that actually is different to the FcRn. There are several other FcRn inhibitors out there, and actually UCB just have had theirs approved for myasthenia gravis. The other inhibitors in this space bind through their Fab arms, which gives them slightly different or quite different cell biological behavior, in fact. But Janssen have one such inhibitor that they actually licensed from or bought from Momenta, and they actually have, I think, ongoing trials in lupus, so we'll wait and see. But I agree entirely that this approach of global depletion is very attractive for diseases where you have a wide array of different specificities, and thank you for your compliments.
Yeah, thank you.
Greg?
Sally, that was very nice, and but like you, I kind of are worried very slightly about the effect of possibly immunizing again against this autoantigen. You know it's a problem anyway, and then you're shoving more of it in a possibly slightly multimerized form on cell surfaces, and the ability that could say stimulate yet further, like a vaccine really. And so you mentioned actually you have some kind of feeling that by being delivered through the liver it could be tolerogenic. But presumably you must know, you should be able to do experiments in mice to show whether it's tolerogenic or not. In other words, to show that, for example, repeated uses of this therapy, in fact, clears these antibodies for good.
Yeah, no, and Greg, those points are very well taken, and actually due to the move and COVID and stuff, we've been slow, but we are doing those studies. Okay. That's something, so that's certainly on our radar list, because completely agreed, we have to de-risk this. You know, we're very excited by the approach, but we have to de-risk it by doing those kinds of studies. So that's...
Well, I mean, it'd be very exciting if indeed it also turns out to be tolerogenic, because it might be lots of other ways in which you can induce that. It's really
Sure. Thanks, Greg.
Sally, hi, that's me. What is the limit of half-life extension? Can you go above the 100 hours?
That's limited by, as soon as you start creeping up with the binding at near neutral pH, basically, you tend to not get so much release of the antibody back into the circulation, and you turn into what amounts to a half-life extending molecule into an FcRn blocker. You know, so what limits that is it's an engineering problem, actually, I think, because there's some point that you actually can't enhance any more at pH 6 and still retain negligible binding at pH 7.4. So that's as I say, what the limitation is, because you need this. And some people, even actually in MedImmune, William Dallowak and his colleagues had a nice paper looking at what's the threshold for binding at pH 7.4 to really still get reasonable release and half-life extension. And they claimed the number was about 300 nanomolar, actually, the affinity. So that's what, as I say, it's just basically the biophysics of the interaction or molecular nature of the interaction that you can't get sufficient binding at pH 6 to go above a certain half-life extension without introducing pH 7.4, which mitigates the enhancement, the effect of the enhancement at pH 6. So.
Cool. Hi. Okay, so we move on.
Okay,
Thank you, Sally.
Thank you.
We now go to, can you please? Thank you. We move now to the oral presentation. It's by Stefano Agliardi. He's a clinician from Milan, working most of his time in the clinic, treating patients. And it's very nice that he's able to present to us some very interesting work about women, pregnant women vaccinated with COVID. So it's all yours.
Thank you very much for the presentation. Good evening, everybody. It's a pleasure to be here. And yes, today I'm going to talk about mRNA vaccination during pregnancy against COVID-19 and the impact of vaccination, of maternal vaccination on the newborns. As you know, at the end of 2020, the very last days, the all great vaccination campaign started throughout all the Western world, and we gave the priority to the patients at higher risk of severe disease and complications and mortality in case of COVID infection. For example, oldest old, immunocompromised, but also pregnancy is demonstrated to be a risk factor for COVID complication. But pregnant women were excluded for the vaccination, for the vaccine pivotal trial, registrational trials, and so there were no data, and also there were no clear indication from national health institutions. So, long story short, it led to vaccination hesitancy. I worked in a vaccination center, and I felt this hesitancy in many people, but in particular among pregnant women. At our center, we tried to develop a sort of task force to collect different specialists and to try to give the best advice to the pregnant couples about COVID vaccination. Firstly, it was more difficult, but luckily later we understood that mRNA vaccines are safe if done during pregnancy. There are a lot of reports. I don't want to waste your time because, of course, you have read them. But still today, after 2.5 years, there are still no data about what happens. Okay, say the woman, I got my vaccination, I produce my own antibodies, but what does it happens to my baby? Does he or she receive the antibodies through the placenta when he or she is in my belly? And which is the amount of this passing? Do this antibody protect him or protect her, and which is the durability of these antibodies? And so, to better understand this aspect, we tried to set up this study. We included pregnant women having received mRNA vaccination during pregnancy, and we performed a serological evaluation through blood samples in these women and in the respective newborns at birth, and we also performed the follow-up at 3, 6, and 9 months. At last, we included 98 pregnant women. Most of them have completed the vaccination cycle with 2 doses. At the time, there were no booster doses. And most of them having received the last dose in the last part of the pregnancy. We also included 180 babies, 103 because we have 5 twin pregnancies, and most of these newborns were received breastfeeding during the follow-up. During the follow-up, we have some lost, some drop out, but only for personal reasons, so nothing significant about, nothing medically significant. And this is what we saw in our analysis. As you see in the upper chart, I have divided the 2 groups among 1 dose group and 2 doses group, according to the number of doses received by the mother during pregnancy. As you see, in the one-dose group, there is a wider distribution, the orange squares. You can have high values, but also low values of antibodies, and the mean is located right in the middle of the chart, and this is both for the mother and for the newborn. In the two doses group, the blue dots are located almost all of them in the upper right part of the graphic. So that means more doses, more antibodies, and you can also see it in the numbers. You can guess that there also a correlation, a positive correlation between maternal and neonatal antibody titer. And you can also see it in the two charts below that are very, very similar. You can also see that there's a negative correlation between days from the last dose from delivery and titers at the time of birth, both for mothers and both for the children. And this is even clearer in these two charts, the B and the 1, where aggregating the population according to trimesters of vaccination, you see that if the women had received the vaccination in the second or third trimester, they and their babies had at birth higher antibody titers. I don't know if we will have to receive new boosters in the future, but maybe this is a thing that we can consider in the case. And at last, I want you to show the more, according to me, which are the most interesting data, the data about the follow-up. You see that in time, there's a drop of the antibodies in the blood of the newborns. The mean drop of our population is the gray dashed line. But you see that this drop is not linear, so we wonder why, and we get the answer that maybe there were a confounding factor, and the confounding factor are the 34 COVID cases that we registered during the follow-up. Luckily, we, among these 34 babies, registered no hospitalization. I don't know if it's due to maternal antibodies, I hope so, but I cannot say that. I just report it to you. But if we split the line, excluding these babies who got infected, you see that the orange line, if you don't get the vaccination, the drop is quite linear. But the babies who got infected, the antibodies that are dropping between 3 and 6 months, where there was a new wave of COVID, started to soar. So that what's that the possible meaning of that is that even the youngest babies at between 3 and 6 months of age may have immune system that is able to respond of infection, and so they are able to produce their own antibodies. This is also the age when all of the babies start to receive their vaccination. So this is confirms that at this age the immune system is active and can protect the babies in case of infection with new antibodies. And at last, let me show the data about breastfeeding. We wondered if breastfeeding had a role in recharging the antibodies among these babies. And among our population, we see that the drop is quite similar. But it seems that among breastfed baby at 9 months, the drop seems to be slowly lower. They seem to maintain more antibodies. I don't know if it has a clinical significance, because we anyway have an important drop of antibodies, but we promote breastfeeding for many reasons. This can be one more reason to consider to promote breastfeeding. So in conclusion, we saw that higher number of doses, more doses means more antibodies that completely reasonable. Sometimes the antibody titers of the babies are equal to or even higher than the mother titers. There is also a correlation between gestational age at vaccination and the serological titers at birth. As I told you, maybe this can be considered if we have to propose a booster to pregnant women. There's also a possible response and an increase in the antibody titers among the youngest in case of infection. And what we told about the breastfeeding, a possible protective role in maintaining higher antibody titers in the first months of life. At last, as a clinical pharmacologist, let me tell that it's not easy when there is a lack of data to treat and to convince and to talk with patients. So we should make an effort to ensure not to exclude the special population from the first phases of research, because this is crucial to ensure an equal, full and safe access to all the patients and to the people that are in need of taking their drugs. Because the patient we study often is not the patient we treat. At last, let me thank all the colleagues that were involved in the study, but in particular all the patients, the mothers, the families, and the babies. These are three of our twins. It's been very good to work in a setting like that. And at last, let me thank you for your attention. If you have any question, I'm here. Otherwise, I give you my contacts if you want to contact me. Thank you very much.
Thank you, Stefano. It was really very interesting. Any question from the audience? Okay, I'll ask a question. I'm sorry, maybe I missed it, but how do you measure the level of antibody with the infant?
Yes, the unit of measures is arbitrary units on milliliters. Is the common serological test that we always use even in adult people through blood samples, a simple blood withdrawal.
Okay, thank you.
My question was kind of following on from that in a way. It's that that's measuring the total amount of antibody.
Yes.
Some predefined thing. But you don't have any hints as to the affinity of the antibodies. It could be lots of very weak affinity antibodies, and did you actually have you got any insight into that?
Yes, you are totally right. But at our hospital, it was not possible to analyze this aspect, and we could only report this kind of data.
Thank you very much.
You're welcome. Thank you too. Good evening.
Okay, I will introduce the next speaker now, Rene Hoet. He will talk about new technology development for future multi-specific antibody discovery and optimization. Rene is the Chief Innovation Officer in FairBiology and Professor of Biopharmaceutics in University of Maastricht. Since 2022. He graduated in Nijmegen, Netherlands. He was a postdoc in Sergey Winter Lab. Then he switched to career in the industry. He was in Dyax for some years, where he had 4 antibody approved for FDA. Then he moved to Genmark and Bayer. He stayed in Bayer for 8 years. And then he joined FairBiology. All yours.
Thank you very much, Ahuva, and thanks for inviting me to this meeting. So what I would like to do today with you is actually talk about antibodies. We already had a whole session about antibodies, so what I would like to go with you through is actually the current challenges of antibodies, because there still are. First, a short introduction to FairJourney Biologics, what we do and what technologies we have. And then I want to focus on a kind of new antibody library we call Explorer, a new modular antibody library, which actually uses 2 technologies, phage display and mammalian display. And why mammalian display? I talk about this also a bit more in detail. Mammalian display is a technology actually to select for the antibodies that are not just specific and high affinity and potent, but also can be developed into therapeutics. So, they can be produced at high amounts, very high concentrations, which is nowadays very important because more and more antibodies are used subcutaneously in patients. And then therefore there's more requirements on the quality of the antibodies for, as drugs. And finally, I end up with some future antibody discovery platform vision at FairJourney Biologics. So FairJourney Biologics is an antibody company, basically, which is a spin-off of Applinks, a Belgium company, which is now part of Sanofi. Started in Porto, Portugal, still based there. Started about 10 years ago. What we basically do is we basically support companies to develop antibodies from the early stage to develop new targets, as well as to select and discover antibodies, engineer them, optimize them, humanize them, produce them, look for functional activity, developability, and also make stable cell lines. So really support the track basically of antibodies to the clinic. From the work of FairJourney, but also from Iontas, but because I didn't tell you, but Iontas is a company which is founded by John McCafferty in Cambridge, and this is now part of FairJourney Biologics. From both companies together, now we know at least there are 14 antibodies in the clinic from those antibodies. We made more than 4,000 antibody libraries in this time, make antibodies from humans, llama, chicken, rabbits, mice, and rabbits. There's now more than 200 people, and we have done 800 projects in this 10 years. So what is the technology about? You heard already a lot from Sir Greg Winter about this technology. The core technology is actually a phage display technology, making libraries of non-immune individuals. On the left-hand side, you see this from healthy donors. We actually also make libraries from llamas. And these llamas, they have a particular single domain antibodies that are smaller, heavy chain fragments, and they are very useful for the construction of, for example, bispecific and multispecific antibodies. So that's one source and to make antibodies. Very often to, let's say, more challenging targets, like, for instance, GPCRs or ion channels, you need immunization to get good antibodies. And we actually have these 5 animals to basically make antibodies. We actually make, after immunization and see that there's immune response, we make antibody libraries from these animals. And these antibody libraries, they're made from basically single domain antibodies, single chains, FAPs, or basically including the Fc, all in principle and phage, and then selected for the targets. And then finally, on the right-hand side, you see actually the new antibody library, semi-synthetic or explorer library, which basically is actually designed on partly taking the immune repertoire from human donors and partly synthetically designed with really the aim to mimic as close as possible the human diversity and also try to improve the biophysical properties of antibodies so they can be easily transferred into drugs. So as I mentioned already, we use as core technology phage display, but also mammalian display technology, and I will tell you more in the rest of the talk also about this technology. So we take antibody discovery as a kind of holistic approach, different species of animals which can immunize, or humans where we have full antibody libraries. As platforms, use phage display, mammalian display, non-immune libraries, but also lately we actually use directly the B cells. The advantage of the B cells is that you keep actually the heavy and light chains of the original B cell intact, and you can directly also get then these original heavy and light chain pairing I'm not going to talk about this today, but there's also a whole new technology direction into the single B-cell technology. So today I'm going to focus on this Explorer library and the mammalian display technology. So I want to go back, at least more than 15 years, describe some of the first semi-synthetic libraries which were made at Diex when I was working there. It's now part of Takeda. And we made actually an antibody library, and 15 years later, we can see actually that from that single library, there's now 4 antibodies approved. Not actually by Diex, only one antibody was actually developed originally by Diex, a P-kallikrein antibody, you see at the bottom, which is actually approved in 2018 by Takeda. But actually antibodies developed by other companies like Lilly and Serono Pfizer. And there's still more than 10 antibodies in the clinic. So this is more than 15 years later after this library was made. So just illustrating that from a single antibody platform, you can have really multiple drugs. And I think that's one of the beauties of phage display technology, have given so many antibody libraries, antibody drugs. The concept, what we actually took at that time, was actually similar as the Explorer library, and that's why I show it. Some part actually we take from human donors, and particularly the heavy chain CDR3 is actually a region of the molecule which varies enormously in sequence, in length between 4 and 30 amino acids, and the sequence is almost completely random. So that's we take actually from human donors. The CDR1/2 we actually designed targeted diversity, not really randomly, but really mimicking already at that time the diversity seen in humans. And we actually used one single framework, which is well expressed. And so that was actually the library we made, and this was licensed to over 30 different companies. So now the Explorer library. So what did we take up with the Explorer library, and what did we improve? So basically, we start with selecting all the very stable scaffolds for the heavy chain and light chain, and first looking at the frequency of these heavy and light chain scaffolds in human repertoires. And then we included the most frequently used heavy chain scaffolds, and we focused on the VH1 and 3 family. We used both the most frequently lambda light chains and kappa light chain scaffolds, and of course also looked that these antibody sequences can be expressed well in phage display antibody libraries from our own internal data. Of course, it's not only important that they are expressed in bacteria, but also in mammalian systems. And we also looked that these frameworks which we selected are present in currently approved therapeutic antibodies, so they're clinically validated. So on the right-hand side of the slide, you see the diversity, what we put in. Part we took actually synthetically, CDR1 and 2 of both heavy and light chains. We actually took basically synthetically mimicked the repertoire which is in humans, and CDR3 heavy chain and light chain which actually took from human donors. And then because nowadays you can actually design so nicely these synthetic diversity, we actually tried to reduce as much as possible the biophysical liabilities in the design. For example, we avoided N-glycosylation and deamidation there. So this was then assembled into an antibody library, which a size typically of over 10 to the 10. And I think the beauty, what we added to the system is actually that we actually have a modular system where we can actually change very quickly to mammalian display, selecting full antibodies or bispecific antibodies in the final format to optimize the molecule. So this is basically the different modules which are made, and it's a kind of plug-and-play system. So you have basically the synthetic CDR3 on the left-hand side of the slide for the VH, CDR1/2, the donor CDR3 diversity of the heavy chain, and then for the light chain, also synthetic and donor diversity. We actually emphasize that we put most of the diversity in the CDR3 of the heavy chain because that's such an important region for the specificity of the antibodies. And so, in principle, it's a modular system, and we can actually use those modules. We have DNA stored of those modules, so we can actually easily go back to those modules to actually optimize the molecules in mammalian display. So a little bit more detail on how basically this, the design was done of this mutagenesis. And I've taken here a CDR1 as an example of the VH3 family member, VH330. So we basically try to mimic as close as possible, as I mentioned, the human diversity. And that means that at some positions in the CDR, we actually left the, we didn't put any mutations there. The original germline sequence for this is, for instance, serine, tyrosine, alanine, methionine, and histidine. And at position 2, you see the tyrosine, and at position 4, the methionine, we actually kept really constant. So moderate amino acids, for instance, the serine at position 1, we put 18 amino acids. So moderate positions, only 5 or 16, really dependent on the millions of sequences which are nowadays already available in the database to look at. So from all these combinations of the selected scaffold, we actually ended up with 20 sublibraries in total, with a diversity of over 10 to the 10. And we actually made sure that the quality of these libraries was very high. They went actually to a hyperphage selection to make sure that there's only, let's say, members in that also express actually antibody fragments on the surface of bacteriophages. And then, of course, we looked at the diversity. Nowadays, with NGS sequencing, you can actually very nicely analyze this, showing some example here of the VH1 family on the left-hand side, and on the right-hand side is a VH3 family, CDR1 and CDR2. We actually, and I forgot to mention this, we actually tried to reduce the number of mutations we put in the different antibodies by only allowing 2 mutations per CDR. So in CDR1, in one member, there's only 2 mutations. That is true for CDR1 and CDR2. And this is to avoid that there is actually too many issues with developability of antibodies, and that there is no biophysical liabilities. And it's also mimicking actually the diversity what happens in humans. Of course, the CDR3 of the heavy chain is important, and typically the curves on the left-hand side you see in the literature just showing the huge diversity in length and distribution of the CDR3. What you nowadays with NGS, of course, can also look is really how many CDR3s are really in this library, and this is just a simple NGS of 90,000 sequences. You see 80,000 are actually only once in the library, and there's about 4,000 that are twice. So this really goes logarithmically down, showing that there is a really excellent diversity in the CDR3 of the heavy chain. So we started validation. I don't really have the whole validation data yet, by 2 simple targets, PD1 and HER2, to get just some antibodies, analyzed the diversity, just took 92 single chains, looked at the antibodies, and we saw already for both targets that already in the second round of selections, we got a decent number of positive clones for both targets, HER2 and PD1. And we actually screened some off rates in BioCore for PD1s, which is shown on the right-hand side. We're doing now a full screening and NGS sequencing, and plan to convert these single chains directly to IgG. That's also one of the things we put in place, so that we can go directly from the single chain to IgG in batches, so we can screen thousands of IgGs really early in the process. One of the hurdles, I think, for discovery of antibodies is still to get very early on to functional data, not just affinity data or binding data, but really functional data, particularly for agonistic antibodies. This is still a challenging part. And next step will also be selections on challenging targets, like, for example, GPCRs, which are discussed quite a bit in this conference. So one of the beauties of these Explorer libraries is that we have all these sub-libraries ready. So if we select a binder or a population of binder from the Explorer library, we actually can quickly actually basically clone in the CDR1-2 library of a certain germline or the light chain of a certain germline into these, in this vector. We can select it by phage display, but we can also have the possibility to go directly to mammalian display to optimize the molecule. And this can be done as a full IgG molecule immediately, or, for example, this can also be a bispecific molecule. So there is a very fast way to optimize candidates immediately to the final therapeutic format of choice. So now I want to talk a little bit more about this mammalian display technology and why we think this is actually a useful thing for discovery and optimizing antibodies. So when we actually are looking for antibodies with the desired target product profile, we want to do this as early as possible in discovery. And let's say in the early days of antibody discovery, people try to get a large diversity of antibodies and try to get the best high affinity antibodies, and then later on only found the function. And very often people were disappointed that they were not actually having the function as they were looking for. So really looking for the early on for the functional antibodies. And the other aspect, which is also mentioned here on this slide, is that we're much more aware that we actually have to produce these antibodies in large quantities, but also at very high concentration. So these antibodies need to be developable. Manufacturability is really important. So these things we can actually do with mammalian display. And why mammalian display? So mammalian display, we can actually screen tens of millions of different clones, not just antibody fragments, but full antibodies. And we get information by the use of flow cytometry on expression level, on affinity. We can actually work on an IgG final format, but also we can input bispecifics in, CAR T cells, CAR T selections in, single domain antibody, TCR. So it's much more than just a typical IgG. We screen directly in the production cell types. So we actually, as a standard system, we use HEC293 for this mammalian display technology, but we also can use CHO cells for that. And we have the potential for functional selection. So the challenge of mammalian display technology is actually that we actually want to have only one gene per cell. Similar as with phage display, where there is a coupling between genotype and phenotype. This is not that easy with mammalian display. And therefore, at Iontis, this technology was developed with homologous recombination, which is assisted by nuclease-directed integration. And for this, tail nucleases were used. So this is illustrated basically here in this slide, where actually the homologous recombination occurs in an exon sequence in the AUV locus. And then with the tail nucleases, specific integration is done. This system is actually consists of two promoters here, one for the heavy chain, one for the light chain. And the light chain, the heavy chain is then anchored to a PGF receptor region, which allows display on the surface of mammalian cells. This is all connected with a blastocidin selection gene. So you can actually select cells that are where this construct is stably integrated. And typically about 3 to 5% of the HEC293 cells have stably integrated this in their genome, and these are selected. This is published work in MAPS from several years ago. You can use now this mammalian display similar as with phage display for, and here is just an illustration for affinity maturation of programs. In orange you see the original antibody, and in red the improved antibody. And this is both for mammalian display shown as for phage display. But that's not the major benefit of mammalian display. Mammalian display, this is just to illustrate you can actually use mammalian display also for these purposes. But the major advantage of mammalian display is actually you can select the clones for optimal biophysical properties, for developability. And to illustrate this, I want to go with you through a case study, which is published, the bocacuzumab case study. You might be aware of, bocacuzumab was an antibody developed by Pfizer against PCSK9. It was actually a mouse antibody which was humanized, and then later with phage display improved from 300 picomole to 5 picomole. And this affinity to maturation actually led to multiple what we call the red flags for developability, which was actually published already in 2017 by Jane et al., where they compared 137 clinical stage antibodies and looked at all these developability assays. And they saw that actually there was evidence for self-interaction and low specificity there. And then this antibody failed in the phase 3 clinical trial, which is usually a disaster because this is very expensive, of course, in failing in phase 3 clinical trial for an antibody. About 50% of the patients actually showed an antibody response against this drug. So when we looked at this antibody, here at the bottom in greenish color, the bocacuzumab, and the original humanized antibody, what we saw in our mammalian display system is that the original antibody, the humanized antibody, displayed much higher level on the cell than the bocacuzumab. So that sort of already was for us a sign that there is something not optimal with this bocacuzumab. So the question was, can we actually improve now this antibody and fix basically bocacuzumab by mammalian display? And the answer, of course, is yes. Otherwise, I would not have shown this example. So basically, by looking at the CDRs and the hydrophobic residues, we made a library of the heavy and light chains, a mutagenesis library of about 10 to the 7, and then put that in mammalian display. And then on the left-hand side, you see actually a flow cytometry picture where their selection is done on the y-axis on PCSK9, the target, and on the x-axis on an anti-FC antibody. So you basically select a population with a red square, which is actually highly binding to PCSK9, but also is highly displayed on the surface of mammalian cells, and this red square was actually analyzed in more detail. So with NGS sequencing, and then these antibodies were cloned into IgGs. 88 antibodies were only analyzed, and all of those 88 had much better developability properties than bocacuzumab. And this is sort of illustrated in the table, which is shown in the middle. You see bocacuzumab there, which actually had a really high polyspecificity level. And typically this is measured, it says DNA ELISA level on insulin and DNA. And actually the selected mutant, which only actually differed 2 amino acids, IE mutation and the EEH mutations, though differed only 2 amino acids in the CDR2 of the heavy chain, they showed a dramatically lower polyspecificity. But not only the polyspecificity was done, also the self-aggregation, the method which is AC-Sins, which is developed by Peter Stessier, which is basically what it's doing is on magnetic beads simulating a very high concentration of antibody and looking for aggregation behavior. Actually, also that behavior was actually much better for the improved variants. Also, expression went up threefold from 69 to almost 200 milligram per liter, and diffuse light scattering went down as well. All part of a developability criteria. So then we went one step further, and they basically took the bocacuzumab, but also the improved variant in a proliferation assay using CD4 T cells of selected donors. So there were 10 HLA selected donors taken, D1 to D10, and PBMCs were taken from those donors, and these PBMC CD4 cells were stimulated in the presence of these antibodies. And what you can see there on the right-hand side, you see the dotted lines, and about 2 is a stimulation threshold. And you see that there's many dots of bocacuzumab above this line of 2 stimulation index. We took as a control alemtuzumab, which is known to be a relatively high immunogenic antibody, which is also come out of this test. And as some good behaving antibodies, we took trastuzumab, for example, and bicocuzumab, which also don't show actually this immune response. You see that the improved 2 amino acid mutations selected by mammalian display, they don't show this immunogenicity. There's no or only one donor which shows immunogenicity, which is quite striking. This is still, of course, an in vitro assay. What I find is also more striking even that these mutations, the 2 amino acid mutations, they were not in T-cell epitopes, in classical T-cell epitopes, which are known to sort of be a predictor for immunogenicity. So apparently aggregation behavior and developability is not just the T-cell epitope, but it's also how a protein folds and doesn't aggregate. So in summary, with mammalian display, we can actually fast-track clinical candidates by screening tens of millions of clones, and we actually can repair broken molecules. We try not to do that, this is just an example to show that you can do it. We actually try to use mammalian display early in discovery to select the ones that actually have the right properties. This is a kind of a second study to convince you that actually you can, with mammalian display, select molecules that are developable. And here, this is a collaboration with a large pharma company where, therefore, I cannot show all the data here. But there were 110 CMC stage biologics, meaning that they were in development, preclinical stage biologics, or some actually in the clinic, where the self-association properties were really defined in detail, as shown all these assays done at the previous slide. When you use now flow cytometry of mammalian display, shown on the left-hand side, and divide them into low display, medium display, and high display, and just selecting on binding to Fc, because all these antibodies have different targets. Then actually you can see from the NGS analysis that there's a very high correlation with the ones that are high displaying actually have a really low self-association score, meaning that are very developable antibodies. You can also use a different kind of ranking here, and then in different classes. So class 1 has a score of 0, so they're very developable antibodies would have no problems for developing to the clinic, and the orange ones are in between, and the red ones are really antibodies that you don't want to develop to the clinic. Also here you can see that mammalian display selects out the antibodies which actually have actually the right properties to develop to the clinic. So there was also another technology, and Ahuva and Nissim mentioned this in her introduction, yeast display, which is also actually claimed to select molecules, good antibodies, over bad molecules. And so we did a comparison between mammalian display and yeast display, and took a few antibodies that actually went in the clinic against angiotensin 2 and MEDI912, which is an antibody to NGF, and then took the original parental antibody and the improved antibody, which had better developability properties. And you can see on the left-hand side that in the flow cytometry, the same analysis was done. You can see that the difference between good and bad is actually in mammalian display much stronger than in the yeast display. And so we got the question, so why do you think is this the case? And one of the differences between yeast and mammalian is, of course, the cell wall, which is there in yeast cells, which is not present in mammalian cells. So it took some time there, but basically, at the end of last year, a paper came out which actually showed that the internalization is very likely the driving force why in mammalian cells you get these selections for developability. And we think there's two aspects on it. First of one is the productivity, the speed that the molecule goes to the surface is to do with the production rate of the antibody molecule. And then the bad ones, they are internalized, and that actually is to do with the internalization rate. So the ones that are self-associated and aggregate, they are internalized. So this was very nicely published in the paper from Nature Communication. So we wondered if we can actually also see this in our system. So we took actually the same 1912 MedImmune antibodies, the parental one, which actually had some issues, and then the approved antibody, which had a higher display level on the mammalian cell surface. And what we then did, we actually did add an inhibitor, cytochalasin D, we added here to actually inhibit the internalization, abolish internalization. And then what you can see is actually that you basically, you can, when you block this internalization, you see actually that the parental antibody also displays well on the surface, meaning that the internalization is crucial really to the success of the mammalian display technology. So then, you know, since I have limited time, I want to, but I don't want, I think it's important for you to know this is not just true for IgG molecules developed with VLT screening, but we extend it now to single domain antibodies only, single domain antibodies on Fc, but also importantly to bispecific antibodies. Professor Sally Ward already mentioned this technology, knob-in-hole technology. We use also here a mammalian display to display bispecifics. On one hand, for example, a VHH molecule, on the other hand, a single chain. And you can actually select repertoires in mammalian display with optimized bispecific molecule. That's actually also one of the features where a lot of our clients have issues with selecting bispecific, the optimal bispecific for their application. You can actually also use an Fc fusion protein and a bispecific molecule. It's undisclosed because this is a client program. And we also can actually select for the right CAR-T displayed target, and we use their Yocat cells for that. And that's important also to avoid inotonic signaling in CAR-T cells. So in summary, we can develop basically the antibody developability in different molecular formats and cell types. Just, I think it's also good to mention that the mammalian display, there's now 3 antibodies that actually made it into the clinic. 5 programs were started collaborations actually at Iontas, and 3 of them are now in the clinic. So there are antibodies already in the clinic that use mammalian display for optimization. So then the last slide I want to finalize is actually coming back to the Explorer library, which is actually not just a phage library, but actually combines the beauty of phage display to actually create high diversity, and then use mammalian display to use those modules to optimize your final molecule. So one step we'll be working on, which is shown on the bottom, is actually now to not just display on mammalian cells, which you can select then for developable antibodies, but also take those mammalian cells and then switch the display to soluble expression. And we do this actually in the microfluidics platform, so you can actually from a single mammalian cell move that from soluble expression, and then we're in the stage where we try to set soluble functional screening, so you can immediately go to your functional antibody in a very short time frame. So I think the summary, I think I already mentioned those things. We have a new antibody library, highly developable scaffold. Our mammalian display technology allows to select any antibody molecule, any format. And we think with the Explorer library, we actually can combine mammalian display and phage display to actually fast track, basically to go directly to highly developable lead molecules. And this is, acknowledge all the people at FairJourney and Iontas, and also some of our clients are here on the boat trip on the Douro Valley. We have our own conference, which we organize in September. everybody can actually join online as well, but we actually also have a very nice venue where we invite people, and it's organized by Dennis Burton, a very well-known antibody expert, and happy to invite you for this conference. And with this, I would like to thank and happy to answer any questions.
Thanks, Rene, for very, very interesting talk. Any quick question from the audience? I'll have a quick question for fairness for Gary. Can you comment on the difference between mammalian display and phage display in terms of diversity and size of libraries that you can do?
Yeah, so one of the limitations of mammalian display is currently that the library size is about 10 to the 7. So therefore, we cannot make very large libraries in mammalian display. And we go through the steps, first selecting a repertoire in phage display, and then using for optimization the molecule. We're also, working on a concept that to further expand the diversity in mammalian cells by actually adding some enzyme which expand the diversity in mammalian cells. But that's work for the future.
Thank you. Thanks, Rene. Any more questions? No? So thank you, Rene. I invite Gary for the final talk.
Yeah, so it's a great pleasure to introduce Gary Merry. He's currently VP of Commercial Operations at Twist Biosciences and has been since 2015. Gary's experience in antibody libraries started actually in 2007 at Slonim Biotechnologies, which was extremely successful and acquired in 2010 by Morphosis. He then became a private consultant for about 5 years or so for various biotech companies. And he's going to present today on Twist Bioscience, their anti-COVID antibody programs.
Kind of, sort of. Thank you for coming, the few of you who are here, and thank you to Ahuva for the invitation. As you probably can tell from my accent, I am from around these parts. I did my undergraduate degree here in Glasgow, and my graduate degree, my master's degree in St. Andrews. But I moved to the United States in 1984 and basically have been there since. Can I have a show of audience participation just to keep you awake? And I know that you're all worried about going to the Cayley tonight, which I think starts quite soon, so I won't be long. Who in the audience knows Twist Bioscience? Okay, well, Rene knows it because Rene works for FairJourney, and FairJourney is a very similar company to Twist. In that we both companies make DNA libraries, and we both do discovery projects for outside companies, pharmaceutical companies, some academics. But I would imagine that most of you here don't have the automated technology to take a very large gene mutant library and screen that against the target. I mean, that's millions and millions of dollars of sophisticated equipment. So most of you will be interested in a provider that provides discovery services. And that would be a company like FairJourney or a company like Twist. Some of this, and I do apologize, and I'm in very illustrious company. I think I'm the only person here, apart from my Italian friend, who hasn't worked for Greg Winter, Sir Greg Winter. But I've been in the library business for, about 15 years, Prior to that, I was in the DNA synthesis business, and prior to that, in the protein sequencing business with a company called Applied Biosystems. And so my background is in building DNA. And what's important when you're looking for libraries of DNA, whether they're antibody libraries or other proteins, peptides, if you noticed in Rene's presentation, he had a very small picture of a DNA synthesizer. A DNA synthesizer, they used to come in 3 columns or 4 columns, but essentially you had to stand in front of that machine that was basically a cocktail maker, and you had to press a button that said A or another G, C, and T, and you would build your oligonucleotide synthetically. Well, the technology at Twist is we have, instead of columns to make DNA, 4 columns, we have a silicon wafer. And we can make up to 1 million oligonucleotides in one run. When I first joined the company, I met with the CEO, who's a very inspirational person, and she was telling me about the technology, and I thought perhaps her end goal, her ambition in life, was to cover the Earth's surface in oligonucleotides, because we can make so many. We have a facility in South San Francisco. We have a facility in Portland, Oregon. We have 8, not synthesizers, we don't call them synthesizers, we call them printers. And we can print basically 8 million oligonucleotides up to 400 bases long in one run. A run's about 32 hours. So nowadays we can take an antibody gene, whether single chain or VHH or whatever, and build the CDR1, CDR2, CDR3. With CDR3 we can include length variation for the heavy and light chains. So we can build FAB type antibodies, small antibodies, single chain fragments within a few days, basically. So the idea is that Twist can actually accelerate the production of gene mutant libraries, and hopefully can use those gene mutant libraries to discover pertinent antibodies. And like the technology that Rene was mentioning, because we're synthesizing a library, 1 variant at a time, so we can basically build a library of up to 10 to the 9 variants by building each variant individually at the same time, we can actually exclude all of the undevelopability options and exclude them from the library itself. The libraries we make, I have to say, are sort of display agnostic. We make libraries and we ourselves use phage display. We can make libraries and have done so for Iontis with mammalian display. We make libraries for other companies based in Vermont that use yeast display. And there are still some companies around that use ribosomal display. So our libraries can be used by any display technology. So I won't waste any time. This is just a sales pitch, so I'll get to the point really quickly. But everybody else, and you heard Rene, have one library. If you go to places like Morphosis in the old days, Adimab, so Diax, Zoma, so on and so forth. They have one library. But to us, DNA is free. We make it ourselves. So what we've done, rather than have one library, is we have a collection of libraries. And what you'll see from the COVID results we're going to show you, that comes in handy sometimes. So we know basically the DNA sequence of all the variants. We've built the library ourselves. There's no developability issues. And very quickly we can, everybody talks about our technology as, state of the art or beyond state of the art. We basically can build oligos. Instead of using columns as a solid support, we use, silicon wafers. So we, as a company, are in the business of antibody discovery, but at the same time, we also offer custom libraries. So some people, and I think Rene alluded to, some people like to start small with a very small library, 10 to the 3, 10 to the 4, 5. And then based on the results from that library, we'll expand it to a larger library. Some people will actually start with a really large library. I've oftentimes been asked to make libraries with more variants in the library than there exists molecules in the universe. And then somebody asks you for a 10 to the 27 GPCR library, and you say, how are you displaying it? And they say, phage display. Well, phage display sort of peaks out at 10 to the 9 variants. So, it's quite a sophisticated calculation on how you can identify variants within that library that are specific to your target by only taking a small portion of it. That's a very sophisticated way of doing things. So we discover libraries, we license the libraries. If any of you work for Cancer Research UK, Cancer Research UK is actually licensing our library of libraries, all of them, and we'll use them to work with their collaborators. So the library of libraries, we have, again, to us, DNA is free. We have 20 different libraries, and the idea is that these libraries are off the shelf. When you need them, when you license them and you need them, we'll send you an aliquot. You get on with your phage display, yeast display, or whatever display technology you're interested in. So what we've done at Twist to validate the libraries, to validate the company's technology, is we've actually done a lot of antibody discovery on our own. We are a discovery company. We don't develop antibodies. We look for partners who will develop the antibody. But we strictly make DNA, we make genes, we make oligo pools, we construct combinatorial libraries for custom use, or we have these off-the-shelf naive libraries which are all humanized as much as they can be. And the interesting, and again, I won't dwell on this because unless you work for BioNTech or Moderna, there's probably not that much interest in COVID in the room. So I'll kind of jump through this. But basically, we've developed multiple antibodies against the COVID virus. The number one set were for the spike protein. It was a neutralizing antibody. And the ACE, the anti-ACE2, it was a blocking antibody. Now, what we did, and this is interesting, we had a collaboration with a renowned researcher at Vanderbilt University, Jim Crow, and he was building COVID libraries before COVID had moved outside Italy. Sorry. And he sent one of his top researchers to Italy to get some samples that he could bring back and sequence at Vanderbilt and build a library. The good or the bad thing, depends how you look at this, is that the researcher came back with the samples and he also had contracted COVID. So there was no limitation on how much blood was available for the library. So we helped him with the library. We built our own library. At the same time, we have a Fab library, a full-scale IgG library. And we ran the target, the COVID, against those libraries. And for whatever reason, our CSO, Aaron Sato, decided to include a VHH library. So we have a number of VHH libraries. He included the VHH, which is, a small single domain camelid type library, which currently, I think, because Ablinks, or now Sanofi, in Belgium have got an antibody in the clinic that is a VHH. There's a lot of interest in VHH from a library standpoint at the moment. So he included that, and the results were kind of spectacular. First of all, he generated over 200 high affinity binders from those libraries within 6 weeks, and looked at each individually in 6 weeks. And this was from 6 weeks from the return of the colleague back from Italy. So that gave us a lot to work on, and we looked at the IgGs, we looked at the live virus data, using the IgGs, and we basically got 4 or 5 that were in the low nanomole affinity, which wasn't bad. But what was very interesting is using our VHH libraries, which we tend to build and then shuffle around the CDR1s and 2s and the CDR, well, not in a VHH, sorry. We shuffle around the content, if you like, in the DNA. And very quickly, we can build a library, humanize it, etc. And the results we got from the VHH were actually superior to the result, in terms of affinity, were superior to the results that we got from the Fab or the specific COVID library that we worked on with Vanderbilt. So we looked at, the neutralization of basically the VHH. We looked at, various affinities. We looked at pseudovirus. It all sort of panned out. And here, if anybody has a hamster, make sure your hamster doesn't get COVID, because they tend to lose tremendous weight and then die. So keep your hamsters safe, get them inoculated, maybe. But you can see from the VHHs we introduced that even just 1 milligram per kilogram of hamsters and I guess a kilogram of hamsters is a lot of hamster, we see neutralization of the virus in terms of the little beasties didn't lose weight as they do normally with COVID. So pretty soon, we looked at the VHH leads. We found that there was in fact 2 epitopes on the COVID ACE2 inhibitor that we thought, okay, how do we look at the other variants? Will our VHH, neutralize the other variants? And we looked at the South African variant, seemingly there was a UK variant as well. And in each case, looking at different dose amounts and rates, we neutralized the COVID virus. So that one of the things we did is actually make a bispecific, or a quattro-specific, whatever you want to look at in terms of VHH. But so if it was interfering or interacting with 2 different epitopes, we had 2 different, antibodies on, the VHH. Effectively, we can, neutralize, most of or all of the variants. I can't say all, but there might be others coming that we can. So this is the beauty of, instead of having one gigantic library or a combinatorial library, or if you have specific libraries for specific applications, very quickly you can develop therapeutics, or in our case, we basically have assets that can be licensed or used by others. People always ask me in the years I've been working with libraries, what is a library, Gary? A library is like a giant Blockbuster video store. Do we all remember Blockbuster videos? You used to go and buy video or rent videos. Well, a library is like a giant, Blockbuster. And you could go into Blockbuster and say, oh, I want to rent Spartacus with, whatever his name was. And they say, oh, it's not here. You'll have to come back. Somebody will return it. Well, with a library, it's like, having all of those movies. And you can go in and choose which ones you want. And having multiple libraries is that we have different sections. We have, the rom-com or the action movies, etc., where we specifically build and engineer a library to the target more than we would with a giant, 10 to the 7, 10 to the 8 library. We have a number of different assets. We have a number of different collaborators. This is 2 quotes from Robert Maybury, who is no longer at Takeda, but he thinks we're pretty good at what we do. And Robert Carnahan from Vanderbilt. I don't know whether it was him that went to Italy and contacted COVID. But, Twist is a company, it's 10 years old, over 1,000 employees, based in South San Francisco, and we have representatives here in Europe. We will license you the libraries. We will generate new antibodies for you. We have special software that will build you biosimilars or, optimize your library. We like to collaborate with our customers because our customers are our partners, not just customers. And in a nutshell, that's Twist. I'll be happy to answer any questions. And if there are any questions, we should maybe just go to the pub and I can buy everybody a beer.
Thank you, Gary. This was fantastic. Any question for the people who are still here?
Can I just ask a quick question? So, Gary, can you maybe expand a bit on how you decide which library to use?
Say again?
Can you expand a bit on how you make the decision on which library to use for which target? I mean, is that an AI-informed decision, or how do you actually make that decision?
That's a great question. It used to be, well, first of all, if you're interested mostly in VHHs or you're interested in GPCRs, we have libraries with functionality that helps the discovery of antibodies for those particular things. If you're looking for something that is kind of unknown to you, and this is a bit of a secret, is I would use multiple libraries. I would always use a FAB, I would always use an SCFV, and I would always use a VHH. And, that, all comes with the license.
This is the luxury of somebody who have all these libraries in the same time, huh?
Yeah, we're good at, Ahuva and I met 4 years ago, and she said, Gary, what? She said, I want a library, but I don't have any money. And we eventually came to a conclusion that probably she benefited from, and we had the library designed, we made it, we had it cloned, and that library actually had some issues that we couldn't discover, and the designer couldn't discover, and eventually Ahuva discovered and then found out it was actually a problem introduced by cloning. She is a clever lady. If Ahuva ever, you want some information, she's a bit fast talker when she's giving her presentation, but she's pretty good at what she does. And she worked for Greg Winter as well.
Anyone have more questions? Actually, I want to ask, I'm really curious about it, actually, I'm serious. You mentioned that the potential diversity that you can use is huge, much beyond the ability to have in a library. And you mentioned that you have some colleagues that are still using ribosomal display. I mean, at the antibody meetings, we hardly see anyone using ribosomal display. So can you comment on that?
I think, I'm in a room with, the mothers, the fathers of phage display. Ribosomal display always has been interesting because its throughput can be, if you're really good, you're standing on one leg, you're facing downhill and the wind's behind you. I mean, it has 10 to the 14 throughput. So the bigger the library, 10 to the 14, you can basically interrogate the whole library. So there are still some crazy people, not many, that are interested in that technology because it has higher throughput than phage display.
And is it working for them?
Yeah.
Okay.
Okay, well, thanks, guys. Not many of you showed up, so I want you to all go out and get all your friends and tell them they missed something special.
Thanks for everybody for Gary and for everybody that stayed till the end. And thanks for all the speakers, and thank you very much. Thanks.